Hey guys, welcome to the Creative Biology, and here I am with another series of the lecture on the protein structure. So as we know that there is and four type of the protein structure. So here that basic about this all type of structures we are learning in this video. So stay tuned till the end. So here that primary structure, secondary structure, tertiary structure and quaternary structure I am writing here as according to its complexity. So these lines, that dark part indicates that more complex structure of that proteins, and upon the lighting of this line that indicate the simplicity, or we can say is less complexity of this structure. Okay, now what is the primary structure? Simply that long chain of polypeptide, that amino acid bind with an peptide bond, is known as an primary structure of any protein, right? So if I am taking here an example, so that in that case, assume this molecule or round as an one amino acid, this as an second. So simply a linear chain or linear polypeptide chain. See, this is amino acid, any amino acid. So that type of structure is known as a primary structure of any protein. Okay, but that type of structures are not very much stable, so they form a secondary structure. Now what is the secondary structure? Secondary structure of any proteins have some of the structural formations, like it's have the alpha helical structure, then beta plated sheet, then another that is an turns, that either alpha turns or either beta turns. So in the secondary structures, this primary structure, or we can say is polypeptide chain, will coiled and make some helical or either plated sheet or either turns like structures. So if I am giving an example, so in that case, let's see here that this type of structure is known as an helical structure. Then this type of plated sheet are known as an beta plated sheet, and the turns between these two, that simply this or this, that simple turns are known as either alpha turn or beta turns, that present between this major secondary structure of amino acid. Okay, or we can say protein. So that's all about the secondary structure. Okay, and now what is the tertiary structure? See, this type of the secondary structures will combine with each other with either hydrogen bond or either disulfide bonds, and they make the tertiary protein structure. Now secondary structures only includes the intravalent bond, that only have an attachment of this type of polypeptide chain with an hydrogen bond. But in the tertiary structures, they includes any non-covalent attaching or any non-covalent bond. So in that case, they includes the intra plus intervalent bond. So they also includes intervalent bond. So that as we taken here example, that is a disulfide bond. So they also include that. Okay. So this type of secondary structure will combine and make the tertiary structure of any protein. Now if I am drawing here that see, this is the alpha helical structure, then one turn is present here, and they start to plating a beta plated sheet like structure, and then again fold, turns, and then make an helical structure. So this type of structures of any protein that known as a tertiary structure. Okay, due to this non-covalent attaching. Okay, that's all about it. And now what is the quaternary structure? As we know that this type of the protein structure are present in either cell or either outside the cell. In case if they are surrounded by any particular environment, like here take an example of water, then water loving amino acids are faced outwards, while that water repelled amino acids are face inward in between these structures. So in that case they form quaternary structure, that is a most stable ever structure. So that let's assume here that this big molecule or big round circle as an tertiary structure of any protein, then this type of 2 or 3 or 4 or more than that tertiary structure molecules will combine with each other and make up the final structure of any protein. That type of structure is known as an quaternary structure. Okay, because any tertiary or secondary or primary structures are not stable in the nature. Due to its less complexity, it will easily cleaved by any of the protease enzyme, any of the cleavage enzyme. So to make themselves more strong, more protected, they will convert into the quaternary structure of any protein. Okay, so that's all about this basic about the protein structures, and in the next videos we will one by one learning the all type of the structures. So stay tuned, subscribe to my channel and watch till the end of the series. And if you like this video, please thumbs up and subscribe to my channel, that's Creative Biology. Okay, and now I am putting a graphical view, or we can say the animation on how they will actually forming the structure, or we can say from the primary to quaternary structure. So stay tuned and watch that animation. Welcome to the animation world. As you can see here, this is the primary structure of any proteins, that is a simple chain of amino acid, and this is the helical structure. That pink color shows the helical structure that join with and turns either alpha or beta turns in the white color. This structure shows you the beta plated sheet in the green arrows, and that center molecules is of either any of the ligand or any of the molecules in the final structures. That plated sheet will join with and turns either beta or alpha, and this is the final quaternary structure when this two, that green and blue color tertiary structure are combined, and with one or two ligands and any chelating molecules, they make the final structure, final stable protein structure. Okay, so that's all about it.